Programa de Pós-Graduação em Biologia Celular e de Desenvolvimento, PPG-BCD, foi iniciado em 2009 com os níveis de mestrado e doutorado. Atualmente ele tem recebido o conceito 4 na avaliação do CAPES. O programa ele apresenta caráter inovador, agregando conhecimento das áreas de biologia celular e molecular, genética e biologia do desenvolvimento, combinando diferentes abordagens científicas e metodológicas, o potencial para trabalhar com algas, plantas, animais invertebrados e vertebrados, além de humanos. Além dos conhecimentos da área de biologia celular e desenvolvimento citados, soma-se ao nosso programa outras expertises que diversificam a atuação e incrementam o portfólio dos egressos, bem como enriquecem as publicações científicas. O programa está organizado em 4 linhas de pesquisa. Atualmente, em 2020, são aproximadamente 60 projetos de pesquisa sendo desenvolvidos pelo corpo discente e docente. E o processo seletivo para o ingresso no PPG-BCD ele é inovador e muito acessível, pois vem sendo realizado de forma online desde o ano de 2015.
O PPG-BCD vem realizando e participando da organização de diversos cursos e eventos para os mais variados públicos. Podemos citar o simpósio Karyokinesis, o simpósio de integração dos programas de pós-graduação do CCB, o qual é realizado pelos 11 PPGs do CCB, o curso de verão em Biologia Celular e do Desenvolvimento, voltado para alunos da graduação, e o curso de férias, baseado na vivência das etapas do método científico, que é destinado a alunos do ensino fundamental. O PPG-BCD se destaca em meio às demais PPGs do estado de Santa Catarina, pois é o único PPG da área de avaliação CB1, Ciências Biológicas 1, da CAPES. É importante salientar que a presença do PPG-BCD como único PPG CB1 em Santa Catarina oportunizou a implantação do Profibio, cujo público-alvo são os professores de Biologia do ensino médio. Também no país não existe nenhum programa de pós-graduação em Biologia do Desenvolvimento. O programa contempla uma carência histórica na área, pois há grande necessidade de capacitação de profissionais qualificados na área de biologia celular, molecular e do desenvolvimento.
O PPG-BCD tem demonstrado potencial para atender diferentes demandas técnico-científicas em diferentes setores da sociedade, desde instituições de ensino superior, hospitais, laboratórios que envolvem o uso de tecnologias avançadas e escolas de educação básica. É importante destacar que mais de 95% dos egressos atuam na área, em instituições públicas e privadas, no Brasil e no exterior. Esse número é alcançado devido ao portfólio de diversas expertises adquiridas no PPG-BCD, que oportunizam a rápida inserção em diferentes setores produtivos e tecnológicos da sociedade. A busca por excelência tem tornado o PPG-BCD bastante atrativo, o que é verificado pelo aumento no número de discentes nos últimos anos. Os docentes e discentes do PPG-BCD estão cientes do seu papel na construção de um PPG forte e consolidado e estão envolvidos no alcance das metas com o propósito de atingir as métricas de excelência para tornar o PPG-BCD ainda mais atrativo aos diversos profissionais que desejam completar a sua capacitação acadêmica em um curso referência em ciência, tecnologia e inovação.
Boa tarde, pessoal. Estamos aqui no último dia de Karyokinesis Symposium. Gostaria de agradecer a presença de todos vocês durante esses dias. Foi incrível e não vai ser diferente hoje. Então, eu estou aqui só para dar um recadinho para vocês de que depois da palestra do professor Mariano, a gente vai dar continuidade aqui mesmo, logo em seguida, à nossa cerimônia de encerramento do Karyokinesis. Então, vou chamar aqui uma pessoa para fazer a introdução, a apresentação do professor Mariano para vocês, e depois eu volto com a sessão de perguntas. As perguntas vocês podem deixar no chat, tanto em português, inglês, espanhol, qualquer língua, que aqui a gente está muito chique nesse ano. Então, podem deixar em qualquer língua que a gente vai fazer as perguntas depois para o professor, tá? Então, eu vou chamar aqui o João para participar. João, a palavra é contigo, tá?
Hello everyone, my name is João Victor. I'm a master's degree student in the graduate program in Cell and Developmental Biology at Upstate, and I have the honor to introduce Professor Mariano Sebastian Viapiano. He is graduated in Biological Sciences with a specialization in molecular biology at University of Buenos Aires, have PhD in Biological Science with specialization in Neurosciences on the same university. He was a postdoctoral researcher at University of São Paulo, UFSC, and postdoctoral in Neurobiology Department at Yale University Medical School in USA. He was assistant professor in the Department of Neurological Surgery at Ohio State University, assistant professor in the Department of Neurosurgery at Harvard Medical School, and researcher and director of the Department of Neurosurgery at Brigham and Women's Hospital. He is currently Director of Research at the Brain Tumors Laboratory of the Department of Neuroscience and Physiology at the State University of New York, Upstate Medical School, and Associate Professor in the same department since 2016. Now, Professor, feel free to start your presentation.
Okay, thank you very much. Thank you for that kind introduction, Joao. I hope everyone can see me and everyone can hear me okay. Now, boa tarde. Good afternoon, everyone. Now, you might have just heard from Joao that I am originally from Argentina, and I also spent some time at USP in Sao Paulo many years ago, over 20 years ago. However, I have chosen for today to give this presentation in English rather than Spanish or Portuguese, because meu português está muito enrustido, fez muitos anos since I left USP, and I don't trust myself to give a full lecture in Portuguese anymore. I will try to speak slowly and clearly, and please do not hesitate to make any comments in the chat in English, ask any questions in English, in Portuguese, in Spanish. I understand the three languages very well, but I only trust myself to speak in English these days. So I want to start by thanking the organizers of the symposium. It was a pleasure for me when Joao contacted me early this year, because I learned that there are investigators in Santa Catarina that are interested in understanding more about malignant brain tumors and about the tumor microenvironment, and in particular about the tumor extracellular matrix, which are all topics in which I work. So I really enjoy this opportunity today to show you some of the work that we have been doing in the past few years about how to find new targets and how to target these proteins in the microenvironment of malignant brain tumors. Because I have too many monitors in my computer, let me move some monitors out of the way, and I'm going to, that's it. I'm going to try to start sharing my presentation with you. So the type of cancer that we study in my laboratory are primary brain tumors of the brain and the spinal cord. And the most common primary tumors in the central nervous system are known as gliomas. These are tumors that you probably know have a very poor prognosis with a median survival that is slightly over a year, about 14 to 15 months, and the long-term survival, more than 5 years after diagnosis, is unfortunately very poor. These tumors offer a number of clinical challenges for the oncologist and the surgeon. First of all, there are tumors in the brain. that means that the surgery is very difficult, and in many cases, the resection is incomplete. Second, because they are in the brain, they are protected behind the blood-brain barrier, which means that they are difficult to treat with conventional therapy, for example, chemotherapy. And finally, these tumors are also poorly responsive to immunotherapies, which are novel therapies that are very efficient against other types of solid tumors, but they are not particularly good for brain cancer. Now, in addition to these clinical challenges, malignant brain tumors, malignant gliomas, present a number of biological challenges for scientists that want to study this particular type of cancer and develop therapeutic strategies. For example, there are tumors that are very heterogeneous, which means that in this cartoon that you see here with glioblastoma, which is the most aggressive type of glioma, there are in fact different subpopulations of cells carrying different mutations and having different genetic and epigenetic programs, which means that there is a lot of tumor cell competition and evolution going on, which of course makes glioblastomas a moving target. In addition, these tumors are immunosuppressive. They tend to prevent the immune system from attacking the tumor, and that's why immunotherapies don't work very well against gliomas. They are immune cold, meaning that these tumors don't present a large number of mutations that are useful for the immune system to detect and target the tumor. And finally, as you can see in this cartoon, they are highly invasive, which means that even if a tumor is resected, even if the core of the tumor is completely taken out, there are tumor cells that are going to remain dispersed in the brain, and they are the major cause for recurrence. Now, my laboratory has been studying brain cancer for almost 20 years now, and the main focus of our research has been not only the tumors themselves, the tumor cells themselves, but most importantly, the tumor microenvironment of malignant gliomas. And what do I mean by tumor microenvironment? Well, the microenvironment of a tumor is everything that is inside the tumor that is not the tumor cells. So in the case of malignant gliomas, what this cartoon shows are all the additional components, all the normal cells of the brain that are part of that microenvironment. You not only find the cells at the core of the tumor, but also an invasive cells at the border of the tumor, but also neurons that interact with tumor cells, blood vessels, endothelial cells that interact with tumor cells, astroglia, microglia, which is a type of immune cell, infiltrated immune cells coming from the circulation, oligodendroglia, etc. All these components form the microenvironment of malignant brain cancer. And as I'm going to show you in the next slides, tumors take advantage of this microenvironment. That means that they use the microenvironment to grow, and they use the microenvironment to invade, and they develop a relationship with normal cells in the brain in which tumors take nutrients or take components of the cells or use them in different manners in order to progress. This slide, for example, shows how glioblastomas establish a unique relationship with the microenvironment, with the rest of the brain, in order to invade. These are pathological specimens. These are autopsy specimens. On the left side of the screen, you see a glioblastoma, the most aggressive type of glioma, that has invaded the brain, going from one hemisphere to the other. On the right side, you see a metastasis of a breast cancer. These are very aggressive tumors in the breast, and they can reach the brain. However, they are unable to invade within the brain. They grow like solid masses. This indicates that glioma invasion, one of the properties of malignant gliomas, results from a unique ability of these tumor cells to interact with the microenvironment. And the movie that you might be seeing on the lower part of the screen is a specimen that we imaged several years ago. This is live brain tissue containing a live glioma inside, and we were focusing mostly on the periphery of the tumor just to show how messy the invasion of the tumor is, how much they are trying to escape the core of the tumor. The point that I'm trying to make with this slide is different features of malignant brain tumors, such as invasion, and as I'm going to show you, growth, etc. They do not happen because of the properties of the tumor alone, but because of the interaction that the tumor can develop with the microenvironment, which is the rest of the components in the brain. This is another example of how much brain tumors depend on their microenvironment. This is an experiment that we never published. We implanted mouse tumor cells in wild-type animals, in wild-type black 6 animals, or in a transgenic strain that is called P65 mice. The P65 mice are a transgenic strain that is deficient in a signaling pathway known as NF-kappa B, because they lack a couple of components of the NF-kappa B pathway, in particular the P65 or relay protein, very important for NF-kappa B activation. And because they lack normal NF-kappa B signaling, these animals have deficient inflammatory responses and deficiency in reactive responses. So they don't heal very well, they cannot fight infections very well, and so on and so forth. And when we implanted tumor cells in these animals compared to the wild-type animals, you can see the huge difference in tumor size. The tumors grew normally in wild-type animals. They barely grew in the P65 animals that were unable to defend themselves, providing an inflammatory response. And you can see on the right side of the screen, tumor volume was very different. We investigated this a bit more and found that the reason why the tumors were not growing in the P65 animals is precisely because of this lack of inflammation and reactive responses. In particular, astroglia, which normally surrounds the tumor and forms a scar, a glial scar, astroglia was absent, and the glial scar was absent around these tumors. So this was an interesting lesson for us, because what we learned from this is that the tumor cells need the microenvironment, and they need a microenvironment that has to be permissive for tumor growth. Implanting tumor cells in an environment that is not permissive for tumor growth, even if those tumor cells are mutant and aggressive and they are driven by oncogenes, if the microenvironment does not help with the tumor eventually will not grow or will not invade. And this is what has led the rest of my research over the past decade and a half, which is, okay, if the tumors are taking advantage of the microenvironment and the microenvironment has to support tumor growth, can we find critical targets in the microenvironment? Can we identify critical relationships between the tumor cells and the rest of the environmental components so that we can disrupt those relationships and make the environment not help the tumors anymore? So we want the environment to stop being permissive and supportive for tumor growth and for tumor invasion, hoping that this will allow us to improve anti-tumor therapy. Now, what can we target in the tumor microenvironment? Well, even before I worked in brain cancer, I was a developmental neuroscientist, and I have been working in extracellular matrix in the brain for over 20 years. And I am particularly interested in understanding the composition and the functions of the extracellular matrix in brain cancer. The extracellular matrix, which I'm going to call the ECM for short from now on. The extracellular matrix is, you can think of it as a scaffold, as a support that is around all the tumor cells and all the other normal cells in brain cancer. And it has always been an interesting source of potential targets for us, because our thinking has been, well, if we can target the extracellular matrix with an antibody, a reagent, as you're going to see in a few minutes, well, perhaps we'll be able to target all over the structure of the tumor. We are not targeting a particular relationship between tumors and blood vessels, or tumors and astrocytes, and tumors and neurons, but we are targeting the overall ultrastructure of the tumor, the brain tumor that is growing and invading. And this is an example of why it is important to develop a good targeting strategy against the tumor extracellular matrix. These are experiments that we did with collaborators several years ago, in which we were trying to target tumors in the brain using an oncolytic virus. An oncolytic virus is a virus that enters in tumor cells and kills the tumor cells. And what we did, we engineered a virus to express green fluorescent proteins, so we were able to see how it was infecting the tumor cells. And what you can see in these images is that when we injected the virus alone in the brain, the virus was really unable to penetrate inside the tumor. And this was because the tumor is protected, if you wish, by all these components of the extracellular matrix, fibrillar proteins, polysaccharides, and so on. But when we injected the virus together with an enzyme, chondroitinase, which degrades some of the components of the extracellular matrix, now we had a much better dispersion and penetration of the virus inside the tumor, and a much better efficacy. And when we studied efficacy in animal models, the animals were living much longer. So the point of this slide is to explain that the extracellular matrix that is surrounding the tumor cells and the rest of the cells in the microenvironment is actually an obstacle for successful therapy, and targeting the ECM is a valid and important strategy. And this is an interesting presentation, an interesting paper, that appeared a few years ago, in which investigators compare gene expression in tumors of patients that were dying within a year because of the glioma versus long-term survivors. So long-term survivors in this case were patients that were surviving for several years, usually 5 or more years, and eventually they were succumbing to glioblastoma. And what this study found is that patients that were long-term glioblastoma survivors had significant changes in the type of gene expression in the tumors. And the most important change, and these are the top genes that were differentially expressed, the most important change is that many of the components of the extracellular matrix, all the ones that are highlighted in yellow in here, were decreased. In other words, patients that were long-term glioblastoma survivors had a deficient production of extracellular matrix in those tumors. And this was very exciting to us, even though we have been working in the ECM field for many years, because it supported our rationale, our strategy of targeting and inhibiting, blocking, downregulating, whatever you want to call it, different components of the extracellular matrix in order to make the environment less supportive for the tumors so that we can improve therapy. Now, this takes us to, obviously, the next question, which is, okay, what are we going to target? What is a good target in the tumor extracellular matrix? And what I have shown in this slide are some of the features that I think a good target has to have. And if a target, after a year or two of research, you are not convinced that a target presents these different features, I think that you have to move on, essentially. First, the target, particular protein or glycoprotein of the ECM, has to be tumor-specific, or at least highly enriched in the tumor, meaning that there has to be a lot more of this target in the tumor than in normal brain, so you don't have toxic effects in the brain. And ideally, you don't want this target to be very predominant in other organs. So, for example, targeting collagen is not a very good idea, even though it's a predominant component in the ECM of many tumors, because any targeting strategy is going to have toxic effects in liver, in kidneys, in the lungs, and so on. Second, the target has to be sufficient for tumor malignancy. This means that overexpressing the target has to be sufficient to make the tumor grow and invade more. That way you demonstrate that the target is important. But even more importantly, it has to be necessary, which means that if we downregulate or block this target, now the tumor has to lose the ability to grow and to invade. And finally, ideally, you don't want to discover the wheel. You want to target a protein for which the structure and ideally the function are also known. So we want to focus on targets for which there is some idea of the potential molecular mechanisms. Now, in my laboratory, we use different lines of evidence to decide how we are going to focus on new targets. We analyze the production of extracellular matrix in clinical specimens that we receive from surgery. We have a battery of assays in the lab for tumorigenesis, including tumor growth, tumor invasion, vascularization, and so on. And we can investigate the effect of different ECM proteins in these assays. And finally, a big advantage of the brain cancer field is that these days we have a huge amount of genomic data that is available in public databases that allows us to make a list, if you wish, of potential targets and find if those targets are commonly upregulated in glioblastomas, if they correlate with particular molecular features of the tumors, etc. And these databases include the TCGA, the Cancer Genome Atlas, which is the most important public database containing genomic data for all solid tumors. The IV database, which is a public database, you can check them online, and the IV database is specific for glioblastoma, etc. Now, we have worked over several years with a number of targets, some proteins made by tumor cells, some extracellular matrix proteins made by tumor cells, some of them made by astroglia in the brain, and so on. For the rest of my presentation, I'm going to focus on one particular family, the Fibulin family of extracellular matrix proteins that we have been studying for, I would say, about 12 to 13 years now. So let's start by let me tell you first what the Fibulins do, because this is a family that is not very well known even for people that study extracellular matrix proteins. This cartoon on the left side of the slide is showing a representation of the extracellular matrix scaffold, which is made by a number of fibrillar components. This could be fibrils of collagen, elastin, polysaccharides such as hyaluronic acid, and so on, fibronectin, etc. Now, this scaffold of ECM is attached to the cell surface, the surface of the tumor cells, by a number of receptors, such as integrins and so on. But this attachment is usually not strong enough. So what Fibulins do, these proteins, as you see in the cartoon, I hope you are seeing the mouse in my presentation, these proteins form a clasp, form a bridge. In fact, the name Fibulin comes from the Latin word fibula, that means clasping or binding. What they do is they tighten the interaction of the extracellular matrix scaffold, the fibrils, with the receptors on the cell surface. And in this way, they enhance the adhesion of tumor cells to the ECM. As you can see in the right side of the slide, there are 8 members of the Fibulin family. Some of them have many splicing variants, like Fibulin 1, Fibulin 2, etc. And they have a huge variability of structures. Some of them are very short, as you can see here. Some of them have many different domains and functional motifs. All of them have a common extracellular domain in the carboxy terminus portion of the protein, which is called the Fibulin domain that defines the family. When we started studying these proteins in glioma, first of all, there were not so many fibulins known at that point. And second, we focus on a particular subgroup, the small fibulins that appear here in the middle of the slide, Fibulin 3, Fibulin 4, and Fibulin 5. And the reason why we started studying these three fibulins is because we detected the expression of these fibulins in malignant gliomas, but we couldn't detect them in the brain using Western blot, PCR, which were the techniques that we had at that time. Fibulin 3, 4, and 5, that we call the short fibulins, they are about 50 kilodalton in size, each one. They were absent in normal brain and present in glioblastoma, in different type of gliomas, actually. And this is an image from the first paper that we published on fibulins, where we focus in particular on Fibulin 3 because it was, of the Fibulin family, the most highly upregulated member of the family in different type of gliomas. And the reason why we only did Fibulin 1 to 5 is because 6, 7, and 8 were not even known at that time. So we knew several years ago already that this particular subset of fibulins, and specifically Fibulin 3, were highly upregulated in glioma, and we thought this could be an interesting target because it's absent in normal brain. What is known about this particular protein? So we went to Medline, to PubMed, and we found that there was nothing known about Fibulin 3 in glioma. In fact, there was nothing known about any fibulin in glioma, which was very exciting, but also terrifying, because that meant that we were going to have to figure out everything. We were going to have to figure out the function, but also the mechanism, which is one of the things that I said, ideally you want to know something about the mechanism, and there was nothing known for this particular protein in glioma. And then we found out that even in other solid tumors, there was also almost anything known. So how do you start studying a novel, completely novel target in the extracellular matrix of a tumor? Well, you start by doing what is called a gain of function and loss of function approach. So in a gain of function approach, you start by overexpressing your protein and see what is the effect in the tumor. And in the loss of function approach, you remove the protein and see what is the effect in the tumor. This slide shows, I hope clearly, that the overexpression of Fibulin 3 is sufficient for glioma malignancy because it promotes glioma growth and promotes glioma invasion. In this slide, you see that tumors that were implanted intracranially, that were producing more Fibulin 3, they had bigger volume, they were more diffuse in the borders were breaking apart because there were more invasive cells, and we developed an assay in the laboratory to measure tumor dispersion through three-dimensional scaffolds, and we were able to see that the overexpression of fibulin made those tumors more invasive. So one of the typical features, or one of the desirable features of a good target in the tumor microenvironment, it is sufficient for glioma malignancy. And it took us some time to develop a good strategy to downregulate the protein. There was no CRISPR at that time as a standard method. So we had to develop very good shRNA strategies. And we confirmed that knocking down this protein in intracranial tumors, these are two different tumor models, CNS1, G8, these are invasive tumor models. We demonstrated that we obtain exactly the opposite effect compared to overexpression. So this means that the knockdown of Fibulin 3 was reducing tumor volume, reducing tumor transversal area, etc. It was even increasing apoptosis. It was increasing cell death, in this case, quantified as tunnel-positive cells. It was decreasing tumor invasion. So essentially, we were showing that Fibulin 3 was not only sufficient, but it was, more importantly, necessary for tumor growth and tumor invasion. And if we were downregulating Fibulin 3, we were decreasing these malignant features of the tumor, and as expected, survival of the animals was improved. And this change in survival doesn't seem very big, but I'm not showing the results in here. But when we combine the downregulation of Fibulin 3 with conventional chemotherapy, the efficacy of chemotherapy was actually much better. So we know that Fibulin 3 is present in the tumor, it's not present in the brain. We know that it's sufficient for tumor malignancy. We know that is, more importantly, necessary for tumor malignancy. That leaves us to the next important question. What is the mechanism of Fibulin 3? If we are going to target this protein, we want to target something that we understand and also that we can measure to make sure that we have done the proper specific targeting. Now, we had a number of ways to try to answer this question. Glioblastomas, which is the main type of glioma in which we work in the lab, they are driven, they are pushed forward, they are initiated, in fact, by a relatively small number of changes, a small number of key mutations. For example, mutation in the tumor suppressor P10, mutation in the tumor suppressor NF1, loss or deletion of the tumor suppressor P53. All this leads to a relatively small number of very important signaling mechanisms that are upregulated, such as receptor tyrosine kinases, PI3 kinases, cyclin kinase, etc. And we first investigated some of these mechanisms, but we couldn't see significant changes in signaling caused by Fibulin 3. That meant that Fibulin 3 was modulating or was regulating some of the downstream mechanisms. But as you can see in this slide, there is a lot to try to unpack in here to figure out, because these key initiating mechanisms, they result in a huge network of upregulated genes with many different functions in glioblastoma. And what we wanted to understand is what exactly within this huge network is Fibulin 3 regulating or promoting or enhancing that can explain the protumoral functions of this protein. And the answer, interestingly, didn't come from us just sitting with glioma cells and trying to analyze one mechanism after another, but the answers came from a better understanding of the interactions between the tumor cells and other cells of the microenvironment, and trying to figure out how Fibulin 3 was mediating these effects. For example, one thing that we noticed very quickly when we overexpress Fibulin 3 in brain cancer, in experimental models of cancer, was that those tumors were very hemorrhagic, they were very bloody. And when we did a more formal analysis, we observed that they have a much higher microvascular density, so a higher number of blood vessels per area. And in fact, Fibulin 3, which is not produced by endothelial cells, is only produced by glioma cells. Fibulin 3 was accumulating around the endothelial cells, around blood vessels, and forming this type of fibrils, this type of wrapping. So we thought, well, there is a provascular function of Fibulin 3. Not only Fibulin 3 does something in glioma cells that we are trying to figure out to promote growth and invasion, but it's also modulating some other components of the tumor microenvironment. And in fact, when we knock down Fibulin 3, which is in this experiment on the upper right part of the slide, there was a significant decrease in tumor vascularization, and the tumors are smaller and they are not so nasty looking like this one. They are not hemorrhagic, and vascular density is also decreased a lot. And when we actually study pure purify Fibulin 3 directly on endothelial cells. These are experiments with brain microvascular endothelial cells isolated from the brain. We developed a couple of assays, one to measure the effect of Fibulin 3 on endothelial cell motility, which is an indicator of vessel formation. And then we did conventional assays to study vascular formation. And what we confirmed was what we expected, that Fibulin 3 was promoting vascular formation in the tumor. This gave us a clue about molecular mechanisms, because there are two major mechanisms that promote vascular formation, in particular in vitro. And those are the most common one that many of you probably know, in particular if you work in the cancer field, is vascular endothelial growth factor, or VEGF signaling. This is classical, and many people in cancer try to target tumor vascularization by targeting VEGF signaling. But another one that is not much, is not ignored, but is not given that much importance by people working in the cancer field, is notch signaling. And notch actually is very important. It's a very important mechanism that regulates the formation and the extension of blood vessels in cancer and also in development. Those of you who work in zebrafish, probably, and in particular in zebrafish development, I am sure that you have heard about notch regulation of vascular formation during development. Well, it turns out that this provascular effect of Fibulin 3 was actually very resistant to inhibitors of VEGFR signaling. And that gives us a clue that perhaps the way by which Fibulin 3 was doing this in endothelial cells was by activating notch signaling. So we went back to the bench and we took our endothelial cells and our glioma cells, and we studied if Fibulin 3 was activating notch signaling. And what I'm going to do is I'm going to spare you the biochemistry because it's very boring and it just has a lot of Western blot. But we confirm the first molecular mechanism of Fibulin 3 in glioblastoma, which was a direct activation of notch signaling, which is true both for tumor cells and for endothelial cells in the tumor. And this activation of notch triggers an intracellular cascade that ends up in the expression of certain transcription factors or transcriptional regulators, and in the expression of a number of target genes that we know today are involved in invasion and angiogenesis. So we had some idea of how Fibulin 3 was working, but you see some lines in here that are incomplete and some question marks, and the reason is because we didn't have a full idea how Fibulin 3 was activating notch, even though it was the first time that an ECM protein has been proposed to activate notch signaling. The second clue to fully understand this mechanism of Fibulin 3 came from studies of inflammation. Remember the P65 mice? The P65 mice that I presented at the beginning are animals that lack a functional NF-kappa B pathway, and therefore they cannot develop reactive or inflammatory responses. So we had those animals aside, and we were studying the effect of Fibulin 3 on tumor inflammation and astrocytic reactivity. And we noticed that Fibulin 3, tumors that were overexpressing Fibulin 3, they had a huge upregulation of this protein, P65, which is part of the NF-kappa B pathway, not only in the tumor core, where the tumor cells are, but also in the tumor margin, where astrocytes are surrounding the tumor and trying to form a scar, a glial scar that is trying to contain the tumor. And in fact, when we did a little bit more histology, we found that the addition of Fibulin 3 to astrocytes, or in vivo, the injection of Fibulin 3 or tumors that produce Fibulin 3, were increasing the activation of P65, the activation of NF-kappa B in both in the tumor cells as well as in astrocytes. So again, we went back to the bench and we asked, we know already that Fibulin 3 activates notch signaling. Is it possible that Fibulin 3 also activates NF-kappa B signaling, perhaps in the tumor cells? And using a variety of tumor cell models, we confirmed that Fibulin 3, even purified Fibulin 3 in culture, was sufficient to activate a number of components of the NF-kappa B pathway, which I'm not going to go into detail about this because this is a pathway that has many different moving pieces, and it has canonical and alternative activation, and we demonstrated that Fibulin 3 was driving NF-kappa B signaling pathway in glioma cells. And that gave us, again, another year of biochemistry. It gave us the ability to complete a full understanding of the molecular mechanisms of Fibulin 3. And what we know now, this is where we are today, we know that what Fibulin 3 does is actually activate by direct binding a protease that is on the cell surface. This is an enzyme that cleaves out the proteins on the cell surface of glioma cells and astrocytes and endothelial cells. It's only produced by glioma cells, but it acts on many cells in the microenvironment. And this ADAM17 enzyme, on the one hand, activates Notch, on the other hand, activates a number of intermediary components of the NF-kappa B pathway. All this signaling converges in NF-kappa B, and NF-kappa B is a transcriptional factor, and it regulates the expression of about 150 genes, of which the most important are the ones that I'm showing here. And these genes are the ones that promote tumor growth, tumor invasion, tumor angiogenesis, inflammation, etc. All that kind of mess, inflammatory mess that tumors really like in order to grow and to invade. And we have defined what is called a Fibulin 3 NF-kappa B axis, knowing that if we have high Fibulin 3, we have high inflammation with Notch and NF-kappa B, low Fibulin 3, low inflammation with low Notch and NF-kappa B signaling. Now, from here, this was very good for us because it gave us a full understanding of how Fibulin 3 was working in tumor cells. And now we could start with the really fun part, which is let's try to target Fibulin 3, because now we have a lot of understanding of the mechanism. And if we target Fibulin 3, we know what molecular mechanisms should be inactivated. We can measure those molecular effects of targeting Fibulin 3 as a readout, as a measurement to see if our targeting strategy was efficient or not. So we started testing a number of deletion constructs, a number of constructs of Fibulin 3 lacking different parts of the protein. And after making many deletion constructs, we found that one in particular, this one, Fibulin 3 DSL, which lacks about 25 amino acids, so to speak, was critical because this sequence was necessary for the activation of Notch and NF-kappa B signaling. These images that you see here, I'm not going to describe them because this is all published results, but essentially those are assays that allow us to measure if Fibulin 3, even purified Fibulin 3, is sufficient to activate NF-kappa B and to activate Notch. And we demonstrate that Fibulin 3 activates this pathway, but Fibulin 3 lacking this little sequence of 20-something amino acids completely loses the ability to activate the pathway. So now, not only we have figured out the mechanism for Fibulin 3, but we have also figured out where to target the protein. We know exactly where to target this particular ECM protein. We did some molecular modeling of the protein. This is a fancy cartoon model of Fibulin 3, and the sequence, this 20 amino acid sequence that is at the end terminus of the protein, it was sticking out of the proteins. That means it was very exposed, and it was an ideal region of the protein for us to develop targeting reagents. And in basic science labs, the best type of reagent that we can develop is usually monoclonal antibodies to target, in particular to target large extracellular matrix proteins. So we spent about a year developing a mouse immunization protocol using the target peptide, which is the sequence in red in here. We screened a lot of clones, and we ended up with a very nice monoclonal antibody, MAB428, which I'm going to call anti-Fibulin 3 from now on. In the past couple of years, we actually have made a recombinant version of this antibody will replace the original mouse antibody with a human antibody, and this is now the recombinant antibody that we test in vivo for anti-tumor effect. So what do we do with this antibody? The first thing we did was to make sure that it was detecting what it should detect. In this case, that it was detecting Fibulin 3. This is histology of clinical specimens, normal brain, low-grade glioma, high-grade glioma, with our antibody, and is as good for histology as a commercial antibody, which is the middle column. And more importantly, our antibody detects the unique perivascular location of Fibulin 3, all these fibrils that I showed before, and it has no cross-reactivity with normal brain. So it's a very specific antibody. So we started testing this antibody in animal models, in models in vitro, for some reasons that I will describe later. The effects in vitro of the antibody are not very good. The effects in vivo are very good. And when we implanted tumors, even subcutaneous implantation, which is very simple, when we implanted tumors and started injecting the tumor with this antibody, we saw a good effect, a good anti-tumor effect. So tumor volume was reduced, vascularization was reduced, which we wanted, because that means that we were targeting the effects of Fibulin 3 not only on the tumor cells, but also on the microenvironment. And more importantly, these Westerns and PCRs here, what they show is that these are just Western blot for different markers of NF-kappa B pathway and Notch pathway. What this is showing is that those pathways were being efficiently targeted and disabled and downregulated. So this was demonstrating that our antibody was doing what we wanted to do. It was being specific to target the molecular mechanisms of Fibulin 3. And when we tested this in other tumor models, intracranial models now, the antibody was reducing tumor cell proliferation, which in here is measured by bromodeoxyuridine capture, and it was increasing cell apoptosis, cell death, which in here is measured by phosphohistone histology. And the most important thing is, well, the antibody prolonged the survival of animals carrying these tumors. Not only glioblastoma, these are examples of animals carrying glioblastoma, subcutaneous or intracranial, and treated with our antibody. And in all cases, there was a prolongation of survival, but also the antibody was effective for other tumors that also produce Fibulin 3, such as mesothelioma or renal carcinoma, which actually gave us very good effects. And here is a very interesting and very useful control. We found a type of cancer, colon adenocarcinoma, that does not produce Fibulin 3. These are tumors that do not produce Fibulin 3. And as expected, our antibody didn't do anything, because our antibody is targeting a protein that these adenocarcinomas are not producing. So this was actually a great negative result, because it was demonstrating the specificity of our antibody. Now, in the last couple of slides of my presentation, I'm going to tell you where we are right now, because this is where we started about 2 years ago, and we are still figuring out all the molecular biology. I have shown you the effect of extracellular matrix proteins, such as Fibulin 3, on the tumor cells themselves. It makes tumors bigger and more invasive. On the vascular supply of the tumors, tumor endothelial cells. I have shown you also the effect of extracellular matrix proteins on astrocytes that are forming a reactive layer around the tumor. But I haven't mentioned much about immune system. And these days, everyone working in cancer is also interested about the activation or inactivation of immune responses, because immunotherapy is a very promising approach to target malignant cancers. And one of the reasons why originally we didn't focus much on immune system is because some of our animals are immunocompromised, so that we can implant human tumors, like nude mice, skid mice, etc. But even when those animals don't have T cells, for example, they still have a good activation of macrophages, which are an innate immune defense against injury, insults, infection, or tumors. So we saw when we started treating animals with our antibody, that there was a huge amount of necrosis inside the tumor and an enormous infiltration of macrophages, which in here are detected histologically with the marker IBA1. It's just a protein that is in macrophages. So we thought that this was because the tumors were dying and the macrophages were coming just to do a cleanup, or they were activated by inflammation. And this was happening in the core of the tumor, in the border of the tumor, etc. But more importantly, we noticed that the macrophages that were being attracted to the tumors that we had treated with our anti-Fibulin antibody, they were not expressing certain proteins that macrophages normally express when they infiltrate tumors. For example, arginase. The macrophages that infiltrated control tumors, they were expressing a high amount of arginase. The macrophages that were infiltrating the tumors treated with our anti-Fibulin antibody had no arginase. What was the relevance of that? I'm going to open a quick parenthesis, and I'm going to introduce the concept of macrophage polarization. Some of you working on immunology or working on cancer biology probably know this concept even better than I do. And this is a very simplified 2-minute introduction to macrophage responses in cancer. In tissues, macrophages that are activated by infection or inflammation, they usually acquire a phenotype that is cytotoxic. So they acquire cytotoxic and phagocytic properties. And these properties of macrophages, they have an inflammatory activation that we call M1 state or M1 status, usually allows the macrophages to kill tumor cells. These are the good macrophages, macrophages that we want to come into the tumor in order to attack the tumor cells. However, tumors have evolved to produce a number of signals that actually suppress this inflammatory reaction of macrophages. The tumor cells produce soluble signals such as CSF1, TGF-beta, etc. And these signals change the macrophages that have infiltrated into the tumor from this pro-inflammatory phenotype to this anti-inflammatory phenotype, in which the macrophages kind of help the tumor because they eliminate dead cells and they help formation of blood vessels, and they are immunosuppressive. That means that they even suppress other immune cells. So the M2 macrophages, which are these ones on the left, they are actually tumor promoting, and we really would like to eliminate these macrophages or force the macrophages to go back to the M1 state. Now, these proteins that I have indicated here in blue color and brown color are typical markers, typical proteins that are expressed by M2 and M1 macrophages. And notice one that is here in M2 macrophages is arginase 1. So going one slide back, the macrophages that infiltrate control tumors, they express arginase because the tumors have changed them into this tumor-promoting M2 phenotype, but the macrophages, that's it, the macrophages that have infiltrated the anti-Fibulin-treated tumors, they are still retaining the M1 tumor-killing phenotype because the tumor is no longer able to change them into tumor-promoting. And we demonstrated that even further by treating intracranial tumors with our antibody or a control antibody, and then we collected the tumor, we isolated macrophages from the tumor by cell sorting, and we analyzed gene expression, and we saw that treatment with our anti-Fibulin 3 antibody was upregulating the expression of M1 markers, which is the good markers, the pro-inflammatory anti-tumor markers, and our antibody was down-regulating the M2 markers, which are the bad markers, the M2 that we don't want. So the summary, which is the title of the slide, is that anti-Fibulin 3 antibody promotes an anti-tumor, this one, the M1 macrophage activation. And recently, about a year ago, we started restudying our tumors, and we realized that we had never paid attention to the interaction between Fibulin 3 and macrophages, but we confirmed that, yes, tumors that overexpress Fibulin 3, they have less infiltration of macrophages compared to a tumor in general, and more importantly, those macrophages are more of the M2 type. So in other words, Fibulin 3 is an extracellular matrix protein that, in addition to doing everything that it does on the tumor cells, endothelium, astroglia, etc., it also acts as an immunosuppressive signal because it tries to educate the macrophages to become M2. And we are now figuring out the mechanism. We know that tumor cells express CSF1, which is one of the signals that induce macrophages to acquire the M2 phenotype, and also tumor cells express CD47, which is a protein that blocks activity of macrophages, and we have seen that knocking down Fibulin with siRNA or inhibiting with our antibody removes these immunosuppressive signals. And in fact, in vitro studies, if we add our antibody to glioma cells cultured with macrophages, we enhance the ability of macrophages to attack the tumor cells, meaning that Fibulin 3 is immunosuppressive for the macrophages, and our antibody is not only an antibody against the ECM, but now it becomes a novel way of doing immunotherapy against the tumor. I just want to close by showing that we are expanding these studies. These are just genomic studies where we are currently finding that Fibulin 3 not only correlates with some immunosuppressive signals for macrophages such as CSF1, but in fact Fibulin 3 regulates a number of other immunosuppressive signals that we were completely unaware of. Some of them, like PD-L1 or CD80, are actually important in suppressing not just macrophages, but more importantly in suppressing T cells, which is a very important population of immune cells necessary to attack the tumor, for the immune system to attack the tumor. So we are starting to develop a working model, which is just a cartoon right now, in which Fibulin 3 is able to activate a number of mechanisms in glioma cell, number of signaling mechanisms, and these signals end up producing immunosuppressive signals. This mechanism end up producing immunosuppressive signals that regulate innate immunity, so macrophage response, as well as T cell response. And this is essentially the research where we are advancing right now. So I'm going to close with this slide. And this slide has this beautiful image that appeared in a review in the journal Oncogene a few months ago, in May or June of 2020. And you see that, this was a review about glioblastoma microenvironment, and you see this beautiful drawing of the tumor cells with the macrophages in blue and astrocytes in green, and some circulating cells in there, and neurons and blood vessels. And the whole review talks about glioblastoma microenvironment. But what is the extracellular matrix? Well, the extracellular matrix is just these scratchy lines in the tumor. And the authors think, well, the extracellular matrix is just a scaffold that holds things in the tumor. And the majority of reviews that you can read are going to have either these scaffold, scratchy lines, squiggly lines. What I hope I have shown you over the past 45 minutes, 50 minutes or so, I hope I'm not very much over time, is that the extracellular matrix of glioma is a critical component of the microenvironment, and is much more than a passive scaffold. It contains many components that are signaling, that are driving the tumor malignancy. And extracellular matrix molecules are optimal targets surrounding the tumor cell and they really deserve full-blown strategies to target these components of the tumor. I'm going to close now with one final slide where I actually have to acknowledge all the people that have contributed so much to this project. This picture is my current laboratory, and in particular, I have to recognize my two senior postdocs, Roshini and Somanel, who did all the work in the past 3 years, in particular, all the identification of the relationship between the tumors and the immune system. I also want to thank my collaborators at the State University of New York, where I am right now, at the prior institution where I have been, and collaborators at other institutions. And of course, money is important for our research, so I acknowledge our current funding and prior funding for this research. This picture is a pretty picture, actually, of SUNY Upstate, the State University of New York in Syracuse. We have snow about 6 to 7 months a year, so this is a picture that we get only in the middle of the summer, and this is our building, and I welcome all of you listening to me to please make comments, ask questions, and hopefully one day visit our lab. And I thank you very much for your attention.
Thank you so much, Professor Mariano. And we have actually a lot of questions here, but I would like to take advantage of my position here to make my own question. And first, congratulations about your research. It was really, really great to hear about what you do at SUNY and at your career. And my question is actually regarding your career. Yesterday we had a roundtable discussion about career, and this is something that really caught my attention when I see someone from, particularly from Latin America, working in such great institutes like you have your passage from Yale, Harvard University, now at SUNY Upstate. And I would like to know if you have any words to Brazilian students about that. How was this way to get in such great institutes? So something like that. It's not actually a question, but asking you if you have any word about that. Last year, I had the pleasure to talk with some professors from Yale, me and João. This is how we actually had the opportunity to know you, and talking with Professor Soret. And in this opportunity, I made the same question, because it's really important for us to see people from foreigners getting this space in such great universities. So it's not my question, but I would like to know if you have something to say about that for us.
Sure. I'll try to be brief. And I have to say that in my case in particular, I think I've been very lucky to be in the right place at the right time, similar to what you did when you met with my colleague from Yale last year. I was also very lucky to meet with investigators at Yale when I was at the University of Sao Paulo, and that was what initiated my ability to come here to the United States. I have to say that, of course, I could just say something conventional like, oh, well, you have to work very hard, etc., and that will guarantee your success, but you have to be very lucky as well. One thing that my first advisor here in the United States told me after I arrived here 20 years ago was that one thing that she had been particularly impressed about was about how interested I was in the research that she was doing, and how much I did, in addition to hard work, how much I really wanted to understand the particular models or biological problems that she was studying. And I think this is very important. This is a very important piece of advice I actually give to young postdocs and to graduate students. You have to choose whatever you like to do in science. It doesn't matter the particular topic. And I would say It doesn't even matter the particular laboratory. Of course, trying to go to the best possible laboratories is important. But you have to be very honest about wanting to do research in which you feel very interested, and you want to communicate that interest to your advisor, to your future advisor. In your cover letter, in your resume, even sometimes your resume is not going to have fantastic techniques, because you might come, I come from the University of Buenos Aires at a time when we didn't have any money to do research, and still we had the ability to analyze research, to understand research done by others, and have good conversations with them, tell them how interested we were on this or that particular biological problem. This is what investigators at any big institutions and big universities value a lot, because we can teach methodology in a couple of weeks, but we cannot induce a person, we cannot create interest in a person that doesn't have that interest. So that is the most important thing, is to really be very passionate about wanting to do research in neuroscience, cancer development, sports medicine, whichever you want, and try to understand as much as possible of the problem, the biological, the clinical problem, and tell your future advisor, I really want to go to your laboratory because I'm really very interested in understanding this particular problem, and would you have a position for me? I think that is one of the most important things that has to be very clear when you interact with potential advisors in other institutions.
That's great. That's a great advice. Actually, it's quite inspiring for all of us, showing that this is possible. You just need to, of course, luck, but show your interest. This is great. Thank you so much, Professor. And now I will start with all the comments. And first, we have here Julia Punhal asking, Professor, can I ask in Portuguese? Or?
Yeah, please go ahead. Ask me in Portuguese. I'm going to. This is going to be a mixture. I'm going to answer in English. A couple of words in Portuguese will be mixed up, and some Spanish as well.
Okay, that's
nice question. Thank you very much for asking me. So I'm actually, by education, I'm a developmental neuroscientist. So before I started my career in brain cancer, I worked both in Argentina and Brazil studying brain development. And when I arrived to Yale many years ago to study brain cancer, actually the laboratory where I went in Yale, they were studying extracellular matrix in brain cancer and extracellular matrix during the development of the brain. And one thing that caught my attention is how much malignant glioma cells, they seem to reproduce or to reuse, if you want, mechanisms that are used by glial cells and by neuroblasts during normal brain development. So I was having, at that moment in 2000, 2001, I was having a project doing development of the brain, extracellular matrix in brain development, and I found that I was able to do the same experiments for brain development and for glioma. And I started becoming more and more interested about glioma and thinking, perhaps I can understand better about glioma and brain and development if I study both of them together, and that led me to start focusing more and more in glioma. It is a very abnormal mechanism of development, if you want, the one that these tumors use.
That's nice. Aline Guimarães is asking, first, congratulating for your work, and como alterar a expressão de proteínas da matriz extracelular? Edição gênica é uma opção? Ainda estamos muito longe dessa possibilidade?
I think edição gênica, genetic edition, is one option. It is possible for some extracellular matrix proteins. For other ones, the mechanisms that regulate the expression of these extracellular matrix proteins are still pretty much unknown. So, for example, what regulates the promoter of particular ECM genes, for the vast majority of these genes, is not known. There are one or two or a dozen promoters that are known, but there are over 1,000 ECM proteins, and I think addition is still not an immediate possibility, not that far away in the future, but not very immediate. We need to understand more about the genetic mechanisms that regulate expression of ECM protein.
Here we have a question in English. Thanks for the great lecture. What would you think to be the most important factor in the initial development of a brain tumor? What drives brain cells to become cancerous?
Well, brain cancer, as well as any cancer, is a genetic disease. All cancers are genetic diseases, which means that tumors have to accumulate mutations. You will not have a tumor if you do not have mutations in the genome. You can pursue any other alternative hypothesis that viruses or bacteria or inflammation may initiate tumors, but at the end of the day, a tumor, in order to be self-sustained, in order to maintain itself, it has to accumulate mutations. Now, what are the mechanisms that drive mutagenesis in brain cells? There are a variety. Tumor cells, brain tumor cells, are not particularly exposed to. They are not produced by external factors like lung cancer or stomach cancer, that they are affected by smoking or by chemical products. So I would say, in a way, what drives the initial development of brain tumor is, one, bad luck, because mutations happen. And two, the location. This is where microenvironment is important. The location of those tumors, the initial tumor cells, has to be in the right microenvironment, because if they are located in other portions of the brain, the tumor will not progress. In the case of the human brain and in the case of animal models, the major region where most brain tumors initiate is in the subventricular zone and the subcortical white matter. Astrocytes and glial cells that mutate in other parts of the brain probably will not become cancer.
Gabriela Holman is asking, the results with Fibulin 3 are very exciting. You showed that Fibulin 3 assisted chemotherapy reduced glioblastomas. Do you believe that this could be a potent therapy against glioblastomas?
Absolutely. And in fact, the reason why I introduced our antibody is because it's a way to show how targeting an extracellular matrix protein that most people will only study for purposes of invasion actually can have quite a significant additional effects, a number of additional effects. I think that good targeting, efficient targeting of Fibulin 3 can be combined with chemotherapy, for example, temozolomide chemotherapy and radiation chemotherapy, and that will provide an efficient long-term improvement in efficacy against glioblastoma. And we are going in that direction. Our antibodies are being humanized as we speak. That means that they are being converted into fully human antibodies that can be tested in patients.
Here we have a question from Professor Jason. He's saying, Fibulin 5 is downregulated in several types of cancer. You would also believe in a positive effect of Fibulin 5 overexpression against brain tumors?
So let me see if I understand the question correctly. So the question is if Fibulin 3 overexpression will also be pro-tumorigenic in brain cancer, even though in other cancers it's actually downregulated. This is a great question because it also shows how an extracellular matrix or any component of extracellular matrix by itself is not enough to determine whether that protein is going to be pro or anti-tumorigenic. That is true. Fibulin 5 is downregulated in several types of cancers, and in fact, in those types of cancers, I believe it's in prostate cancer, the model it comes to mind right now, overexpressing the protein is anti-tumoral. And the same is true for Fibulin 3. Fibulin 3 is upregulated in glioblastoma is downregulated in some cancers, and in those cancers, the mechanisms of Fibulin 3 actually reduce tumor formation instead of promote tumor formation. In a way, it's a way to show that individual ECM components they have to interact with the rest of the microenvironment in order to promote tumor growth.
Let me see here, Renato Frank. Há composto promissor contra a fibulina 3 que possa ser usado futuramente em tratamento?
A big problem about targeting extracellular matrix proteins, and I suspect that this question is referring to what we call small molecules, small molecule compounds, molecular pequeñas, not antibodies or large anti-tumor biological agents. A major problem about targeting components of the extracellular matrix is that there are usually no small molecule compounds that are efficient to inhibit, to block the functions of extracellular matrix proteins. And there are a number of reasons, biochemical reasons for this. But basically, small molecules or compostos, they work well on enzymes that have active sites, kinases, phosphatases, etc. And extracellular matrix proteins are very different in structure. So unfortunately, what we need to use for now are big, bulky antibodies, which are blocking of extracellular matrix proteins. But this is a beautiful area and a beautiful line of reasoning. It would be great to develop smaller and smaller compounds against extracellular matrix proteins, so we actually can give these compounds peripherally instead of depending on antibodies that they have to be injected in the tumor site.
Here we have a general question that was almost all answered, and from Julia Cunhal. Quais são os sintomas de risco para esse tumor? O histórico familiar tem muita influência? Existem formas de prevenção?
This is a relatively rare tumor, and I should have actually mentioned this in my presentation. The number of cases in the United States, and for what I remember is very similar in Brazil, is approximately 25 to 30,000 brain tumors in total in the country every year, of which about 15,000 of those are gliomas. So when you make the calculation based on population, total population, the population in Brazil and United States is very similar in the 300 and something million. The incidence of these tumors is rare. So that means two things. One, that we do not know much about risk factors for these tumors. And as long as I know, there are no identified risk factors for glioma. And by risk factors, I mean external risk factors, such as exposure to smoke, exposure to chemicals. I am sure that some of you must have read articles about the use of cell phones, and let me tell you, no, they are not a risk factor for gliomas. They are absolutely not a risk factor for gliomas. Because we don't know about what initiates gliomas, other than, as I said, mutations happen, there are not really good approaches for prevention of these tumors. Whether the historico-familiar has influence or not in these tumors, yes, absolutely. Some few versions of glioma are familiar variants of glioma. Usually they are present in families where there are mutations on P53, the P53 tumor suppressor. But by and large, the vast majority of patients with glioma are spontaneous, and they are not inherited forms of brain cancer.
Gabriela Holman is asking if could Fibulin 3 be efficient in the treatment of other types of tumors? Yes,
absolutely. More than Fibulin 3, the question would be, could anti-Fibulin 3 be efficient? And yes, as you might remember in my presentation, I showed that our anti-Fibulin 3 antibody not only works against glioblastoma, but also works against mesothelioma and also works against renal cancer. In fact, we are right now finishing, it will finish in a month. We have completed a 2-year project demonstrating that anti-Fibulin 3 is a therapeutic agent. The antibody is a therapeutic agent against mesothelioma models, several mesothelioma models, and more importantly, is diagnostic for the patients. And my answer to this question, my general answer is, I believe that anti-Fibulin 3 is a valid strategy for any solid tumor that overexpresses, that has high expression of Fibulin 3, number one, and where Fibulin 3 has been shown to promote the tumor. And I'm sure that there are more tumors than gliomas for this.
That's interesting. Cecilia is asking the relation with Fibulina 3 com IGF-1R, se há relação no contexto de gliomas?
Not that I know of. That's an interesting question, because when we started studying the signaling mechanisms of Fibulin 3, and you might remember the slide in my presentation where I said, well, there are a few key mechanisms that drive gliomas, and one of them is the activation of receptor tyrosine kinases, which are actually all through the roof. They are super activated in glioblastoma cells. Activation of EGF-1R is common in glioblastoma cells, together with activation of EGFR, PDGFR, c-Met receptor, etc., etc. However, we did not find any effect of Fibulin 3 in activating any of these receptors or depending on any of these receptors. That's why we ended up in more complex pathways like Notch or NF-kappa B. So the answer to the question is, I do not know about any relationship between Fibulin 3 and this particular receptor.
Gabriel is asking here, do you know if Fibulin 3 is evolutionarily conserved among the species? I mean, if there is other phyla that could express Fibulin 3?
Fibulin 3 is very highly conserved, at least among mammals, which are the only species that I look at, unfortunately. Human Fibulin 3, mouse Fibulin 3, and rat Fibulin 3 have over 90% homology. The little sequence that I was showing in the N-terminus of the protein that is the target of our antibody is identical between human and mouse and rat, which is great because that means that we can target Fibulin 3 in human glioma cells or mouse glioma cells, etc. But now, moving out of mammals, we know that Fibulin 3 is expressed in other vertebrates. It is expressed in birds, it is expressed in zebrafish, it is expressed in xenopus, and I have at some point, we have made a little bit of an evolutionary mapping of the N-terminal domain of Fibulin 3. Now, interestingly, Fibulin 3, and this is true for many extracellular matrix proteins, are usually conserved or with different levels of conservation in vertebrates. Extracellular matrix in invertebrates, and this is for those of you who study Drosophila or study C. elegans, is very different. It's very, very different compared to the extracellular matrix of vertebrates. And in fact, I do not know about the equivalent of Fibulin 3 for invertebrates. For example, in Drosophila, we were looking for a Fibulin 3 equivalent in Drosophila. We didn't find anything that had significant homology. I mean, I think the protein that we found had less than 15% homology with mammalian Fibulin 3, and we thought, well, that protein really doesn't mean much for us. So yes, I would say that ECM, studies of ECM, in particular in cancer, they are restricted to vertebrates. Models of cancer in C. elegans in Drosophila should not try to focus on ECM because it's going to be very different from what we see in invertebrates.
Professor, I think now we don't have any more questions. Just congratulations and thank you and everything. So I would like to call João here. João, are you ready? Can I put you here so you can say your goodbye to your prospective advisor? Yes. So Professor Mariano, thank you so much. Here you can later read all the thanks here. But I would like to say thank you so much for this opportunity. It was a pleasure having you here. As someone say here in the comments, it's going to be unforgettable for us. So thank you so much.
I thank you. I really had a lot of fun giving this lecture. It's a pleasure for me to interact with students like you. I see a lot of drive, a lot of energy in wanting to do good science, and I'm very happy that you invited me to this symposium, and I'm really happy that I was able to contribute a little bit. Kathleen, thanks so much. Joao, thanks so much also for the invitation.
Thank you, Professor.
Thank you, Professor.
Bye-bye.
It was a great presentation. I appreciate very much. And I have a question, actually.
Go ahead.
About my
really? You
my
have
work.
a question?
My work.
Oh.
So, why did you think about MMP2 and MMP9 as a target for the glioblastoma?
I think they are good targets. I think that there has been an enormous amount of work in targeting those proteases, but I have targeted proteases at some point, and a difficulty in targeting proteases like MMP2 and 9 or other members of the MMP family, etc., is that they usually have many targets themselves. So that means that they cleave, they cut on many components of the extracellular matrix. And that means that targeting the MMP can give you a lot of effects that you won't be able to predict. And also MMP2, MMP9, they are present in other tissues, so you have always the possibility of side effects, like you said, toxicity in other organs, etc. That's why they are good. They are certainly good targets, but the possibility of advancing targeting strategies to the clinic is difficult because of the side effects.
All right. Thank you very much. Thank you for accept the invitation.
Of course. And I hope that we stay in touch. I would really like to know more about your work and about the work of everyone working there in Floripa.
Thank you so much, Professor. Now we are going to let you, I believe you are in lunchtime now.
Say that again, Kathleen? Sorry.
I believe you are in lunchtime now. So.
Yes, it's like 1:30 here, so I think it's fine.
Okay, thank you so much, Professor.
Thank you.
It was a pleasure. Bye.
Bye-bye.
Agora, deixa eu botar a chavezinha para o português. Pessoal, continuem aqui, porque a gente vai dar prosseguimento ao nosso fechamento do evento. Foi realmente, se eu não me engano, foi a Priscila que comentou aqui, se eu achar o projeto, mas realmente aqui, está inesquecível. Foi isso que eu falei para o professor. O evento está inesquecível, mas chegou ao fim, e a gente está muito feliz de chegar nesse momento. A gente vai sentir falta, mas a gente está se sentindo, assim, realizada por ter conseguido fazer isso. Toda a comissão organizadora, né? Então, a gente gostaria de agradecer muito. Tem uma breve apresentação aqui para a gente falar um pouquinho sobre o evento, como que foi tudo. Consegue projetar aí para mim, por favor? Continuem aqui. E em breve vou chamar o professor Jason também para falar com a gente. E tem a premiação também, né? Então, bom, primeiro aqui estão os nossos patrocinadores, que sem eles não seria possível muita coisa. O Recife Design Marketing foi responsável pela nova logo do Karyokinesis Symposium. e pela identidade visual, as nossas cores, algumas artes. Então, muito obrigada. A editora UFSC, a Rio, foram responsáveis por alguns prêmios que a gente vai dar para o pessoal que ganhou, né? Foram selecionados dentre as categorias oral e pôster. E o Vencenciar deu um patrocínio monetário para que a gente conseguisse manter essa plataforma onde a gente está fazendo a nossa live. Então, muito obrigada aos patrocinadores, muito obrigada. Foi, assim, sem vocês não teria sido possível fazer do jeito que a gente fez. Então, muito obrigada. Bom, um geralzão do que foi o nosso Karyokinesis, né? A gente teve... Aliás, Karyokinesis, a gente teve uma grande discussão como é que a gente ia chamar o evento. Eu, particularmente, chamo de Karyokinesis, mas a gente ficou Karyokinesis, Karyokinesis, a gente pode chamar do jeito que a gente quiser. O importante é a ideia do nosso evento, que está se tornando muito importante dentro da nossa PG. A primeira edição já foi espetacular, importante para a PG, comemorando os 10 anos, né? E eu acredito que vai ser um evento que vai ficar para a nossa história. Então, foram 4 palestras, 3 mesas redondas, 9 apresentações orais. Esse ano a gente teve 3 palestrantes internacionais, que foi algo excepcional para a gente, foi uma coisa que a gente queria muito realizar e conseguimos. E 4 palestrantes de outras universidades e institutos brasileiros. Isso dentro de 3 idiomas, então foi muito bacana. E pode passar, por favor. Então, a gente somou dentro do Karyokinesis. A gente teve a infelicidade de não poder fazer isso presencial, mas o evento sendo online possibilitou que a gente alcançasse esses números. 462 participantes. O que são os participantes? São os 394 inscritos que a gente teve e coautores de pôster, de resumos, né? Então, a gente teve ótimos números, a gente conseguiu alcançar muitas pessoas, 40 submissões de resumos no total. E a gente teve dentro do nosso comitê científico de avaliação, foram 24 avaliadores envolvidos. Dentre isso, docentes do programa e também egressos do programa. Então, a gente conseguiu envolver uma força-tarefa muito legal. Pode passar. Aqui os nossos números do YouTube, isso foi até ontem, tá, pessoal? Mas a gente ficou um pouquinho pequeno, mas... A gente teve 3.200 visualizações aqui no YouTube, nos nossos dias de evento até ontem. Hoje a gente cresceu com esse número, com certeza. Tempo de exibição, 1.3 mil horas de evento, o pessoal assistindo o nosso evento. Então, foi muito incrível. E aí, agora, eu vou dizer para vocês, todas as palestras, se vocês verem, a gente tem uma playlist que vai ser o segundo Karyokinesis Symposium. Então, vocês sempre que quiserem reassistir, vai estar sempre disponível, tá? Todas as palestras do evento. Aí, a gente botou aqui uma fotinho, algumas fotos da nossa comissão organizadora. Aquele vídeo ali foi um vídeo que foi a nossa primeira reunião, foi presencial, quando nada disso estava acontecendo, nem a gente nunca ia imaginar que ia acontecer. Então, quando a gente estava começando a planejar o nosso Karyokinesis, a outra foto ali, a superior, que a Júlia está de máscara, foi a nossa primeira reunião virtual, onde a gente decidiu dar prosseguimento ao evento. E vocês podem ver ali a diferença entre a gente no começo e a gente no final. Com certeza tem uma diferença aí, mas foi muito gratificante. Apesar do cansaço, foi muito gratificante. Pode passar. Então, a nossa comissão organizadora, o estudante de mestrado Luan Santin, eu, estudante de mestrado também, a Júlia Punhal, estudante de doutorado, a Gabriela Roman, pós-doc do programa, a Carla Davico, estudante de doutorado, o professor Jason como professor responsável, estudante de doutorado a Júlia, o Tiago também do doutorado, a Aline Guimarães também pós-doc do programa e o estudante de mestrado João Vitor. Então, essa foi a nossa comissão organizadora. E eu aproveito aqui para agradecer a própria comissão organizadora, porque nada disso teria sido possível sem o nosso trabalho em conjunto. Então, muito obrigada, comissão. Foi realmente um prazer trabalhar com vocês e vai ser uma experiência que eu vou levar para o resto da minha vida, com certeza. Bom, a gente gostaria de deixar, então, os nossos agradecimentos à coordenação do Programa de Pós-Graduação em Biologia Celular e do Desenvolvimento, as professoras Evelise Nazari e a professora Yara Muniz, ao comitê científico do evento, a todos os palestrantes que a gente teve o prazer de receber aqui, aos professores docentes do programa que foram moderadores nas nossas mesas redondas, aos autores e coautores dos resumos, muito obrigada por participarem do evento, a todos os participantes, vocês que estão aqui nos assistindo, e, claro, os patrocinadores que nos ajudaram a realizar esse evento. Eu gostaria de deixar aqui, como presidente do Karyokinesis, um agradecimento muito, assim, é até difícil falar, é enorme a minha gratidão para quem eu vou chamar agora, que é a Carla. Carla, não vou te desmutar ainda, porque a gente está aqui na mesma sala, né? Mas, Carla, foi um prazer dividir essa presidência contigo. Ontem a gente falou que a gente estava almoçando, correndo para dar tudo certo, para dar conta de tudo, e tu falou, a gente fez uma boa dupla dentro da presidência, e eu concordo muito, eu acho que deu muito certo, né? E eu te agradeço muito, Carla, por isso, por esse prazer de trabalhar contigo, e eu vou parar de falar, senão eu fico muito emotiva. Vou deixar a palavra contigo, tá, Carla?
Muchas gracias. Voy a hablar en español porque el evento es internacional. Tuvimos 3 idiomas, así que me voy a permitir hablar en español. Muchas gracias, Kathleen. Increíble trabajar con vos, una guerrera de la organización, siempre al frente, haciendo, decidiendo, así que eso y mucho más para todo el grupo de la organización, y también un agradecimiento especial para cada uno que está ahí atrás. Creo que sin audiencia, sin submisiones de trabajo, sin póster, el evento no hubiera existido. Así que muchas gracias a todos por estar ahí, y ahora voy a llamar al profesor Jason para hacer una breve despedida, pero antes decir quiénes son los ganadores seleccionados por el comité científico de mejor póster y mejor presentaciones orales.
Bom, boa tarde a todos vocês. Eu acho que já estamos aqui com adiantado da hora. Por mim, nós poderíamos ter terminado com essa excelente fala da Kathleen e da Carla, mas, claro, enquanto elas preparam aqui o vídeo para a gente expor, então, os ganhadores de cada uma das premiações, eu vou só fazer uns breves agradecimentos aqui. Claro, a Kathleen já comentou de várias pessoas que foram importantes, mas eu, particularmente, não teria como deixar de agradecer a essa comissão de organização do evento, desse nosso segundo Karyokinesis, ou Karyokinese, como queiram. Né, vou nomear aqui as nossas presidentes, a Kathleen e a Carla. Vocês viram também aí a Aline, a Gabriela, durante o evento, né? O pessoal até está brincando aí que a Gabriela achou uma nova profissão aí de youtuber, porque foi muito bem aqui nas moderações, nas apresentações. A Júlia esteve por aqui, o João Vitor apareceu, e ainda tem que agradecer, claro, a Julian, o Luan e o Thiago. Então, relembrando, quando a gente começou a organização desse evento, lá numa época ainda pré-pandemia, eu lembro da Júlia chegar e falar, olha, a gente gostaria que você estivesse conosco, e começou a nomear quem era o time, quem ia fazer parte da organização desse evento. Eu falei, olha, não tenho como, mas eu vou dar um jeito, eu não tenho como negar, porque realmente é uma equipe fantástica. Vocês viram um evento que enfrentou desafios nunca antes imaginados na história. A gente começou fazendo uma organização de algo que era para ser presencial, de repente vem uma pandemia, ninguém sabe se vai poder continuar, se não vai poder continuar, se vamos ter internet suficiente, se não vamos ter. A gente faz ali algumas reuniões online e decide, vamos encarar, vamos tentar esse desafio. Ninguém foi treinado para isso, mas a gente vai tentar e vai dar o nosso melhor. E ao decorrer de todo esse tempo de organização, certamente tenho certeza que essas pessoinhas que eu nomeei aí deram muitas horas do seu tempo, deram muito do seu melhor para fazer esse evento acontecer. Algumas coisas a gente, infelizmente, não controla, né, como a saúde perdemos um palestrante por motivos de saúde aí nos últimos minutos, nas últimas horas antes da apresentação. Tivemos algum outro probleminha de internet, também super aceitável, até mesmo porque nós tivemos aqui palestrantes da Argentina, do Brasil, de várias regiões do Brasil, da Espanha, de Portugal, dos Estados Unidos. Então, foi realmente um evento internacional em 3 línguas, como as meninas colocaram, né? Então, fica aqui meu super agradecimento para essa comissão. Realmente, o evento foi um sucesso, não só pelas visualizações, pela vitória em todos os desafios que apareceram aí de última hora ou de antemão, mas que, com certeza, a gente cumpriu o objetivo principal do Karyokinesis, que seria também o objetivo se o evento fosse presencial, que é de dividir e multiplicar conhecimentos nas nossas áreas de biologia celular, biologia molecular e biologia do desenvolvimento. Como vocês viram no videozinho, que passou várias vezes aí nas aberturas das palestras, esse é o único programa de biologia do desenvolvimento, na sua essência mesmo, em todo o Brasil. Então, aproveitem para conhecê-lo no futuro, presencialmente, ou mesmo entrar em contato aí durante essa pandemia com todas essas meninas e meninos que fizeram esse simpósio acontecer, para tirar dúvidas, para saber mais como é fazer uma pós-graduação aqui, quais laboratórios existem, quais são os orientadores e assim por diante. Elas e eu e eles estaremos todos aí sempre presentes para responder essas dúvidas de vocês. Eu vou deixar também um agradecimento especial para a coordenação do programa de pós-graduação em Biologia Celular e do Desenvolvimento. Elas foram citadas já aqui, a professora Evelise Nazari e a sua subcoordenadora Yara Muniz. Elas estiveram presentes aí no chat ao longo de todo o evento, congratulando o pessoal e fazendo perguntas específicas. Certamente essa é uma dupla de coordenação que funciona tão bem como a Kathleen e a Carla, a melhor dupla possível que talvez a gente tenha para organizar um evento aqui na pós-graduação é a Kathleen e a Carla, e talvez a melhor dupla possível que talvez a universidade nossa tenha para coordenar um programa de pós-graduação seja a Evelise e a Yara. Elas têm realmente um efeito de sinergia ali quando estão próximas as duas e propiciam coisas muito fantásticas. E, claro, eu vou deixar aqui também, estender um pouquinho mais os agradecimentos, então, para todos os nossos patrocinadores que fizeram isso possível, para todos os palestrantes que dispuseram do seu tempo, como o professor agora no horário de almoço, o professor Mariano, aqui 2 horas, basicamente, com a gente, respondendo um monte de perguntas, trazendo ciência de altíssima qualidade para todos nós, e assim foram os vários os exemplos. Posso citar a professora de Portugal, que terminou aqui com nós quase meia-noite, a professora Tamara, lá da Espanha, no horário de trabalho dela. Então, sensacional ter contado com todos aqui. A abertura, onde a gente teve um probleminha lá com obras específicas, a professora tentando se virar ali no momento para resolver. Foi realmente muito fantástico. Os palestrantes fizeram desse evento sensacional, ele maior do que a gente imaginava ainda, mais sensacional do que a gente imaginava. Agradecer a todos vocês que estiveram aí do outro lado, ressaltar aquilo que a Kathleen já colocou, que as palestras estarão gravadas, disponíveis para quem quiser reassistir, tirar alguma dúvida específica. Então, se inscrevam aí no canal do Karyokinesis no YouTube, deixem o like ali no vídeo para ele ser impulsionado para mais pessoas, para que ele possa atingir mais pessoas, a gente consiga divulgar aí os nossos trabalhos para muito mais gente do que a gente já conseguiu durante o período específico do evento. E, gente, então, acho que, infelizmente, vai deixar saudade, mas em algum momento a gente tem que encerrar. Então, eu vou chamar as meninas aqui de volta, acho que já está tudo pronto para a gente passar aqui o videozinho, agora anunciando os vencedores, os vitoriosos em cada uma das categorias, nesse segundo simpósio Karyokinesis. Meninas, com vocês.
Obrigada, professor. Então, pessoal, vai ser um videozinho muito rápido e eu volto aqui para dar um tchau final. E é isso, confiram o vídeo. Quem foram os ganhadores? Então tá, pessoal, vocês viram aí quem foram os ganhadores. Então a gente vai entrar em contato com cada um para combinar a entrega dos prêmios, os prêmios que a gente tem graças aos nossos patrocinadores. Então eu vou deixar aqui minha despedida, gente. Muito obrigada por tudo. E foi incrível e vai deixar saudade.
Muito obrigada a todos, novamente, agora sim em português. E então, já vamos entrar em contato com os ganhadores. E muito obrigada até as pessoas que estão ainda aí, online, assistindo cada uma das palestras. Muito obrigada. Gratidão.
Bom, pessoal, então é isso. Encerramos aqui o nosso segundo simpósio. Espero vê-los futuramente, tomara de forma presencial, aí pela universidade, nos encontrando na universidade, no nosso programa de pós-graduação. E já fica o convite aqui para, de repente, estarem presentes aí também no terceiro simpósio no ano que vem. Um grande abraço para vocês todos e até breve.